Chapter 13, Natural Selection and Adaptation. All right, so now we're getting into Unit 4, which if you remember at the beginning of the course, the way that it's set up, we're set up kind of like an ahupua'a, right? So we went from the sea, where we're learning about sustainability, malama aina, malama ikakai. We went up into the land. We're learning more about the building blocks of life, starting with cells and working our way up, and that was in the kalo fields. And then we just covered genetics. You guys ended with your genealogy, mo'oku'auhau, and now we're kind of up at the top. So we're looking over everything that we've learned already. We're looking at how everything's kind of interconnected, and we're ending with evolution. Okay, so we're discussing how life evolved on Earth, and we're going to do it a little bit in the context of the Kumulipo, which is the Hawaiian creation chant, just because obviously it follows how life evolved. And we'll get more into evolution in a few chapters, but we're going to start with natural selection. So it's kind of branching genetics and evolution together. So we're kind of working our way slowly up the mountain before we get full on into evolution. But we'll start with natural selection, adaptation, and go into that before we get into how life actually evolved. Okay, so again, natural selection first, we'll end with human evolution. So by the end of this chapter, chapter 13, you should understand the mechanisms behind MRSA infections, which is what your book talks mostly about, or it focuses this chapter on MRSA and different antibiotic resistant strains of staph. Okay, be able to define evolution and fitness, explain natural selection and adaptation, and know the 3 patterns of natural selection. Okay, so before we get into what your book goes over, which is again more the antibiotic resistant drugs or superbugs, I should say, how do populations actually evolve? So you've heard of evolution and maybe you don't quite understand what it is or not yet, but that's what we're going to get into. So how does a population actually evolve? Well, for a population to evolve, a few things have to be true. Okay, you have to have a genetically diverse population, meaning that you have to have different alleles in the population. Organisms need to look different, need to have different phenotypes or different genotypes. Sorry, they will also probably have different phenotypes. Okay. Also need a varying allele frequency. So remember, each gene has, for our sakes, two alleles. So we need to have varying allele frequency of what alleles we see in the population. And the environment has to favor some alleles over others. Okay. So evolution is what that change is. So as allele frequency changes over time, that's what's called evolution. Okay. So say we have this genotype. We have a heterozygote, capital A and then lowercase a. And, you know, these are our 2 alleles in the population, let's say. If that changes, so let's say we start off with it's 50-50. We have 50% of big A, 50% of little a, right? And then over time, eventually, our dominant one, now we have 75%, and little a, we've only got 25%. That is evolution. Okay, so our allele frequency is changing over time. Keep in mind, evolution is not quick, right? Evolution usually takes place over really large amounts of time. It's not something that you will usually see in your lifetime. You might start seeing hints of it, but it's not something that you can study and see within a generation or even a few generations usually with longer lived organisms. You can potentially see evolution of something that can divide really quickly and have lots of generations in a short period of time, like bacteria, which is why we talk about that so heavily in this chapter. So when we're talking about evolution, one of the things you'll commonly hear is fitness. And a fitness is an organism's ability to survive and reproduce in a particular environment. So you want high fitness, right? The higher fitness you have means the increased likelihood of alleles being passed down to the next generation. So organisms with higher fitness are the ones that are more likely to mate, or even if they don't mate, if they just reproduce asexually, they're the ones more likely to pass down the alleles that they have to their offspring. Okay, so when we're talking about these superbugs in this chapter that have antibiotic resistance, the reason why they're so prevalent is because those alleles that confer the resistance to the antibiotics are increasing that organism's fitness. So if they're increasing the organism's fitness, it's more likely that they're going to be the ones that survive and that they're going to be the ones that reproduce. So I'm sure you've also heard of natural selection. Natural selection and evolution can sometimes be used interchangeably. Remember, evolution is the change in allele frequency over time. Natural selection is, you can have evolution as a product of natural selection, or yeah. So natural selection is differential survival and reproduction of individuals within a population in response to environmental pressure. Okay. So this is when you have advantageous traits that become more common, and then therefore the population becomes better suited or more adapted to the environment they're living in. So this natural selection and adaptation both rely really heavily on the environment that you're in. So when we're talking about these superbugs in the chapter, these alleles for antibiotic resistance might not be the ones that infer higher fitness and be the ones that are passed down necessarily. It depends heavily on their environment. Since they're in an environment with lots of antibiotics, we have a problem all over the world, in America as well, of overprescribing antibiotics and also not taking the full round of antibiotics, that we're exposing these bacteria, basically these prokaryotic cells, to way too much antibiotics. And so because of that environment, they're becoming better adapted to resist those. Okay, so we've talked a lot about these bacteria. So what actually is it? The one that we talk about most in this chapter is methicillin-resistant Staphylococcus aureus, or staph, as it's commonly referred to. Methicillin-resistant staph is specifically called MRSA. And you might have heard about it before. But basically, it's an infectious bacterium. It's very difficult to treat with antibiotics, and it can kill you. It kills a decent amount of people every year, about 19,000 people in the US each year. So it's been around since the 60s, and if you remember when we talked about antibiotics, I think in our second unit for the first time, we talked about penicillin, our wonder drug, right? That came about in the 1940s, killed a whole bunch of bacteria. We mass produced it for war, and it was great. But all of a sudden, 10 years later, we were having penicillin-resistant bacteria, right? Because we had too many antibiotics. Those populations had evolved or adapted to that environment of having so much antibiotics and developed or evolved resistance. Okay. By 1961, we had a new antibiotic called methicillin. And there's lots of different penicillin, methicillin, vancomycin, lots of different sort of penicillin-derived antibiotics. And the most common ones are called beta-lactams, which we'll talk about here in just a second. But staph, or S. aureus, mostly hear about it called staph. A lot of strains are harmless, and a lot of ones cause disease, so it just depends. Most of us, about 40% of us, have staph on our nose or on our skin, and it's not really an issue usually unless it gets into your bloodstream. So if you have an open wound or a cut, that's when it gets in there. And I know some of these pictures are pretty gnarly, but things like using soap, towels, equipment, contaminated surfaces, so like locker rooms are real breeding grounds for this. So you see a lot of athletes that have this sort of problem because of the locker room situation and skin-to-skin contact with other people. Sometimes it's pretty mild infection, sometimes it's really severe. It just depends. And most of us have probably had a staph infection, and it might not have been bad enough that you had to do something about it, and it might have been pretty bad. I had one time, I had a dog, this was like 15 years ago, and I had jeans on, and my dog jumped up on my leg and actually scratched through my jeans just because she gripped onto my leg so hard. And that cut in my leg actually ended up getting staph because I probably got some bacteria from her nail as well. And I didn't know I was cut so bad because I was wearing jeans. Who would have known? So that was a bad staph infection. I had to get some antibiotics and some shots for it, basically. So those things happen, and hopefully most of the time they're not really too dangerous, but it is something that you want to keep track of. If you start to see infectious areas like this, definitely go get treated for it immediately, because especially if your immune system is already weakened, it can be a real problem. So don't let this scare you too much, but definitely be aware that these exist and that it can be fatal, or it can cause a lot of other issues as well. Okay, and the ones that we're really concerned about, obviously, are the drug-resistant strains. So MRSA came about because it developed resistance to antibiotic drugs. And again, it does adversely affect people with weakened immune systems. This is one of the examples your book gives. It is a very sad story about an athlete, Ricky Linetti, who was already weakened by the flu, and basically it progressed really fast. I think within a day or so, he had really low blood pressure. I think he had some cardiac issues too. They couldn't really figure out what was going on. And then eventually, well, really quickly, he passed away, I think within a day. And then eventually, when they got the autopsy, they found out that it was through MRSA. It was a MRSA infection that was basically able to take over his body a lot more easily because he was weakened by the flu. So most of these strains are also, we call them MRSA a lot of times, but they're actually also VRSA, which means that they're resistant to vancomycin. And vancomycin is the strongest penicillin that we have so far discovered. So if it's resistant to vancomycin, which I think Ricky's story, they had given him rounds of vancomycin and he was not reacting to it whatsoever. So that was a very powerful VRSA that he was dealing with, or MRSA. Uh, Unfortunately, we kind of use these interchangeably, but VRSA is a little bit different. If you can't kill off the bacteria with vancomycin, it would be considered VRSA. So if you remember back to antibiotics, what are they? What are some of their functions? Again, a lot of the times we use these things called beta-lactams. Those are most commonly prescribed. And basically what it does is it kills bacteria, or it can sometimes slow their growth depending on which beta-lactam you're using, or if you're using a different antibiotic. But usually what happens is it's interfering with the function of the cell wall. Okay, so it'll interfere with the synthesis of the cell wall so that it's not able to synthesize cell walls, basically, and then water's going to flow into that cell by osmosis, and then the wall is going to break apart and the cell is going to rupture. Okay, so that's ideally what we want, right? But once we start using too many antibiotics, if we're prescribing them for things that don't have to do with bacterial infections, if we're not taking our full rounds, that's when we get bacteria that emerge that survive the antibiotics. So this is why it's really important, if you're prescribed 2 weeks of antibiotics, you take all full 2 weeks or 3 days, whatever it is that you're prescribed. Because if you take only a little bit of it and you start to feel better and you're like, I don't need to do the whole thing, what happens is you've given your body a little dose of that antibiotic, right? So you've killed off some bacteria, definitely. But the ones that have survived have now been exposed to that antibiotic. They've survived it, and now they have a higher fitness and are more likely to, one, survive, and two, pass it on to the next generation. Okay. So antibiotic resistance can happen in two basic ways, right? So you've got to acquire these new alleles and bacteria that have antibiotic resistance, and that can happen through either random mutation or something called gene transfer. Okay, so like I said a little bit earlier, bacteria are asexually reproducing, which we'll talk about in just a second. So that means that they can reproduce very quickly. So you can see bacterial evolution in a relatively short time period. And when I say short time period, you're not going to see this in a few hours necessarily, although you might see genetic changes. You might see allele frequency changing a little bit in that time period. But like when we're talking about introducing penicillin in the 40s and then having 10 years later, lots of different resistant strains of bacteria, that's kind of what we're talking about. So this time scale is very short compared to most evolution timescales, which are thousands of years usually, if not millions. Okay, so again, this can happen through either mutations, and that's mutations made, or basically mistakes during DNA replication that introduce new alleles into the population, or something called gene transfer. And gene transfer is literally exactly what it sounds like. You're passing DNA from one bacterial species to another. Okay, so the vertical transfer is what we refer to as the mutations, right? So this is going straight up and down, basically. So you can acquire these random mutations when DNA replicates during asexual reproduction. So bacteria go through something called binary fission, and that's basically when one parental cell turns into two daughter cells. So similar to mitosis, right? We're not undergoing meiosis with bacterial cells because they don't have sex cells. They don't reproduce sexually. So they're doing this asexually. Basically, the cell's going to elongate, the DNA is going to be replicated. They're just going to make one big massive cell and then split it down the middle. So we're going to go from one bacterial cell into two daughter cells. Okay. So that's the first way that that happens. And I should mention that asexual reproduction, binary fission in bacteria, again, can happen very, very quickly. So you can see generations of bacteria within 20 minutes because it can happen so quickly. And because it can happen so quickly, that also means that you have greater chance of having more mutations. So, you know, we don't have too many mutations. We might have one in, let's say, a million, for, you know, 1 in a million base pairs that happens, but our DNA is not replicating that quickly. We're not going through um, sexual reproduction. We're not, you know, none of that is happening as quickly in, let's say, humans or even a lot of other organisms. But bacteria, all of this is very fast, which means that they can evolve much quicker, they can adapt much quicker, and you can see these mutations arise much quicker. Okay, the other way is horizontal transfer. So this is that gene transfer we were talking about. And again, we're just passing DNA directly back and forth between each other. It doesn't even have to be the same species. You can be crossing a non-staph bacteria with a staph bacteria. So you can be giving, let's say, a non-staph bacteria that has antibiotic resistance, can hand that over to a staph bacteria, and they're like, okay, here's some antibiotic resistance. We know you love that, right? It's going to increase your fitness. You're going to live longer, and you're going to be able to kill off anything that you're in. So they can pass those back and forth to each other across different bacterial species. So that is another way that we can confer antibiotic resistance. Okay, so you'll remember from our last unit that the alleles or your genotype, so let's say if you're heterozygote, right, that determines your phenotype, okay? Not the other way around. So genotype is determining phenotype. There is an interplay between phenotype and the environment that determine the frequency of traits in a population. So again, remember the environment plays a big role. You wouldn't necessarily be well adapted to an environment all the time. Like you might think of an organism that would be really well adapted, like a polar bear, to the Arctic. But just because it's well adapted to that environment doesn't mean that if you put it somewhere on the equator that it would do well. It would likely have very low fitness if you put it into that environment. Okay, so if the traits are improving the fitness, then the traits become more common, right? Trait becomes more common, the population evolves, our allele frequencies are changing over time, and that's what we refer to as natural selection. Okay, so natural selection again has a lot to do with the environment that the organisms are in. Keep in mind that individuals do not evolve. Okay, you might have mutations that arise in your DNA, think of like humans, let's say, but you're not evolving over time. Okay, so we're evolving again over large amounts of time, but we're also evolving as a population. So just because you have a mutation in you, let's say, or even in your family, you're still evolving as a population, not an individual. Okay, so these populations are experiencing changes in allele frequencies over time. Our allele frequencies as individuals don't really change. Again, you might have some mutation, but a lot of times that's at birth. Okay, so natural selection does occur in patterns, and there's 3 different types of natural selection that we'll talk about. The first is directional selection. So just what it sounds like, directional. The predominant phenotype is going to shift in a particular direction. The next is stabilizing selection. So this one is near the middle of the range. And then we also have diversifying selection, where you have both extremes of the range that we're selecting for. So let's go over it in a little bit more detail here. So directional selection, this occurs when a single phenotype predominates in a particular environment. So the example here is antibiotic-containing environments that favor resistant strains of bacteria. So you can see the number of bacteria here, and then we have antibiotic resistance. You can see the blue is before natural selection. So you can see we have low low antibiotic resistance before the alleles that confer antibiotic resistance come into play. So as we get more of those antibiotic resistant alleles after natural selection, you can see that our antibiotic resistance is high so that we can select for that phenotype. Another good example that your book gives is, I think, tan and black mice. So say you're in an environment like a desert and you have tan and black mice. Which mice would probably do really well in that environment? Probably the tan ones, right? Think about a tan mouse on light sand or brown dirt versus a black mouse. The black mouse is going to stick out a lot more, and so they're going to be more likely to be predated on, versus the lighter mouse will probably do a lot better. But let's say that we have a shift. So we go from this desert environment, a volcano erupts, and now we have all black lava. Right? If we have all that black lava now, the tan mice are probably not going to do as well. So now all of a sudden their fitness has changed. Where they were really fit before in the environment that they had, now their fitness has changed because now they stick out like sore thumbs on this black lava rock, whereas the black mice are going to start doing really well. They're going to start surviving longer, and then they're going to be able to reproduce and produce more offspring that will have those alleles that will go through natural selection and change over time or evolve over time. Okay, the next one is stabilizing selection. So this is where we go right into the middle. And a good example here is human babies. And when you think about babies, right, you don't want a really small baby. Babies with low birth weights, especially like premature babies, don't survive nearly as well as larger babies do. A lot of times there's different health risks associated with them as well. But on the other hand, you also don't want a really large baby for probably multiple reasons, but especially as the mom, makes it really difficult to deliver through the birth canal, right? So in this case, we actually prefer those mid-range babies. Those are the ones that are favored. So that's usually what we end up that's mostly what our population has evolved. You meet a lot more people who were born in, let's say the 5 to 8 pound range versus the ones that are under 5 pounds and the ones that are over 8 pounds, right? We don't want the really little or the really big babies. We want right in the middle. That's our stabilizing selection. Okay. And last but not least, we have diversifying selection. So this is kind of when you have a patchy sort of environment and you favor the extremes of the phenotypic range. Okay. So as an example, we have finches. So let's say that we only have medium seeds available on some island, right? And we've got small and large finch beak sizes, right? Or well, let's say we have small, medium, and large, okay? But we only have, actually I said this backwards. Okay, so we only have small, medium, and large finches, and we only have small and large seeds. So the small birds are going to be able to eat all those small seeds really well. The large birds are going to be able to eat the large seeds really well. But the medium birds are going to have a difficult time eating the small seeds. You know, their beak might be too large to pick at them or open them up, whatever it might be. And they're also going to have a hard time with the large seeds, right? So in this case, we're actually favoring both the small and the large. We're favoring both extremes of the range, whereas those medium finches aren't going to do as well because they don't have food that that they would prefer and that they would be better at eating. Okay, so that's diversifying selection. So how do we stop superbugs? How do we help someone who has MRSA or VRSA, right? And I mean, to some extent, it is what it is. We've never discovered anything stronger than vancomycin as a penicillin type drug that can stop that. And, you know, a big part of it is we've got to be able to control resistance by changing practices that enable resistant strains to thrive. So we need to stop prescribing so many antibiotics. We need to make sure we take the full round of antibiotics. We need to wash hands. The answer for everything seems to be wash your hands, use hand sanitizers, um, basic things, right? Especially if you're an athlete or if you're in the gym a lot, in the locker rooms, if you have contact with people, always wash your hands, your body. Make sure you don't have open wounds when you're in contact with other people, or, even open wounds if you're going into bodies of water or anything like that. Okay. There are a few other ways that this happens, right? Again, with athletes, it's very common, so you need to be able to keep locker rooms and sport equipments clean, make sure you're washing your hands. Do not take antibiotics for viral infections, right? If you have a virus, do not if your doctor prescribes you antibiotics, tell them no. Tell them you have a virus, you do not have a bacterial infection, and therefore antibiotics are not going to help you, okay? Research vaccines. In recent years, there's been a big anti-vax push. And again, not meaning to get political on you, definitely do your own research. I'm not saying that every vaccine is great, although I'm not saying that. But vaccines do prevent resistant bacterial strains from making people ill. They prevent lots of different things. We're probably going to have a vaccine for COVID soon. And that's a scary thing when these things are new, but they are reasons that they're there, and herd immunity is a very real and important thing that we need. So I challenge you, if you've heard of any, or if you've been leaning towards anti-vaccinations, to please do your research, and again, look at the science behind it. There are no actual scientists that will tell you to stay away from vaccines. Okay. Another big problem that we have is we give excessive antibiotics to livestock. So like say your chicken, this is why you'll see um, chicken and beef advertised sometimes is that they don't give them antibiotics, or they're hormone free, all sorts of different things, right? Because that's another way that we're getting antibiotics into our system, is if we're eating these animals. The other thing is that sometimes if they have lots of antibiotics in their feed, it's leaching into the water as well, and we're getting that through different ways too. Okay, so that's another common issue. Okay, so that's all about natural selection, and starting to get into evolution and how it all works, how organisms become better adapted to specific environments. And then we'll get into the next chapter, getting into Hardy-Weinberg equilibrium, how to tell if a population is in Hardy-Weinberg equilibrium. And that's probably one of the most difficult concepts of this unit. I do recommend reading Milestone 5, The Adventures in Evolution, to read about, I think it probably has Darwin and Wallace, in their sort of battle that wasn't really a battle between them, but it's more so a battle between us.